everybody with the Med13L organization. It's really an amazing group of people, and I've been now involved with them for quite a number of years, actually quite a number of years. And so we got into this sort of by chance, but once we got into it, we've not turned back yet. We've kept at it. Yeah, exactly. She wouldn't let me. Does Vanessa scare anybody else? I don't know, man. So anyway, so with the phenotypes associated, the symptoms associated with Med13L, it may not be too surprising that not only does Med13L have an important role in the nucleus, controlling gene expression, as we just heard so nicely discussed, but we also found a sort of an indirect role for Med13L in the cytoplasm involved in mitochondrial function. So what I want to do today is basically explain some of the characteristics of mitochondria. I'm not touching this, I'm really not. How about if I just hold it? Yeah, I'll just hold it. Okay, is that better? So what we want to do what I want to do today is explain some of the characteristics and activities of mitochondria and make it more understandable why we're so focused on this aspect of Med13L function and to disease-related conditions. A postdoc in my lab, Alicia, who many of you know, will be talking about this this afternoon in not one, but two talks. Please give like an introduction to the details she'll handle this afternoon. So everybody remembers from their 9th grade biology class that mitochondria are? All right, well done, everybody. I knew that you would get it. So one of the things that's associated with that 9th grade biology book is a EM picture of mitochondria. As you can see here, they look like little beans. And one of the things about this, though, I want to point out is these cristae, these little lines here, these are invaginations in the mitochondria, and this is where the ATP is generated in these things right here. And it's actually important for mitochondria not to be individual beans like this. As a matter of fact, if you look, they don't actually look like this. And if you see what mitochondria really look like in a cell, this is actually an image I made. These are cells of fibroblasts. And the mitochondria are elongated. And they have these very specific, they go out through the entire cytoplasm. This area here is the nucleus, but you can see how the mitochondria go out and become elongated. And that's actually important. And the shape of the mitochondria is going to be important for some of the functions it has. And what I want to do is talk about that shapes affect mitochondrial function. And I got this out of order, so I'm going to go here first. So this is a mouse embryonic fibroblast, so it's a typical cell that we use in the lab. And you can see, like I pointed out earlier, the mitochondria are elongated. Most of them are. And in general, this is the normal way you see mitochondria. But also you see them like this sometimes too. And this is the hyperfragmented version of it. It's very clear to see, right, how this is so much different than that. But under normal conditions, when we grow the cells, this elongated mitochondria is really important because it minimizes ATP generation, so the energy part. It also helps store calcium. It also helps store iron to keep it out of the cell. It also allows mitochondrial DNA. So the mitochondria have their own DNA. So it's not just the nucleus. Mitochondria have their own DNA. And allows the mitochondrial DNA and the lipids to repair themselves if they're elongated. Now, there are certain situations where the cell switches to this fragmented version. And the one that people recognize the most is in response to cell damage. So cells can be damaged by a variety of mechanisms. One of the more important ones, especially for what we're interested in, is reactive oxygen species, or ROS. And this is reactive oxygen, which will modify lipids and proteins, and DNA will damage those things. And if you have enough damage, you go through something called mitochondrial outer membrane permeability. We call it MOMP, okay? So everyone can call it MOMP. And this leads to programmed cell death, or regulated cell death, or apoptosis. So this is a very common feature when you get this fragmentation in response to cytotoxic drugs. However, mitochondria will fragment under other conditions as well. And one of them is during mitotic cell division. Just when cells divide, the mitochondria will go into this fragmented state because it's easier to segregate them into the daughters. So you don't want to try to pull these big, long mitochondria around. You just want these little small ones to be able to segregate into the daughter cells. And the second time mitochondria will fragment is under a process called mitophagy, which is sort of a quality control mechanism that mitochondria have. When you get damaged mitochondria, they start leaking this ROS, this reactive oxygen, and that's detrimental to the cell. And mitophagy is a way of identifying those damaged mitochondria and degrading them through a process through autophagy, okay, is the overall thing. So anyway, so you've got mitochondria fragmenting when the cells want to die. You've got mitochondria fragmenting when the cells want to live. And you've got mitochondria fragmenting when the cells want to get rid of some dysfunctional mitochondria. And the problem with this is that the same protein, this protein called DRP1, is involved in mitochondrial fission in all three cases. So how does the cell decide? How does it do that? And so what I want to do today is talk about these different types of cell division or mitochondrial fragmentation and how they relate to neurodegenerative diseases and things that we're interested in the lab. So let me go back to this. And so the first thing I want to talk about is the partitioning, when you just fragment the mitochondria to allow them to be easily moved. And this is a neuron, okay? And this is a picture of the neuron body here, and then it goes through these axons to the synaptic regions out here. And these little yellow dots are mitochondria. And it turns out that in the synapses, you have to have mitochondria out there because it's a very energy-dependent process as neurons connect. But the problem is these axons can be really long. And so the only way to get mitochondria out there is to move them along a little trail that they make with these protein motors called kinesins, which will take the mitochondria out to the synapses. Now, it's been shown in yeast and other organisms too as well, is that this trip that they take works the best if the mitochondria are functional. They are more likely to make it to the end of the trail here. And if they're not functional, they tend to remain in the primary body here. So having mitochondrial fragmentation happen here to allow these small mitochondria to move to the ends is very critical. But the fact that the mitochondria need to be functional is also important because you don't pull dysfunctional mitochondria out here. Only the functional ones can go. So that would be one aspect of how you want mitochondrial fragmentation to help you. I got to go through here again, sorry. And so that we go back to the question now, so how does the cell decide that this is a damaging mitochondria induced fission or this is a mitotic division mitochondrial fragmentation? So again, this is a former graduate student in the lab. And again, these are MEF cells, and the blue dot here is the nucleus. This is DAPI staining, and here's cyclin C in the nucleus, as a good transcription factor should be. And here's the mitochondria are elongated. So if you stress these cells with a pro-oxidant, you get mitochondrial fragmentation. But you get this interesting thing here is that some, but not all, of cyclin C goes to the mitochondria. Most of it stays in the nucleus, but it does go out here and form these little puncta bodies out here. And if you look at this carefully, you look at the overlay, you see cyclin C overlays with the mitochondrial fragmentation. And so we subsequently used this data. We did more studies. We found that cyclin C actually directly interacts with DRP1, this fission protein, and allows this mitochondrial fragmentation to happen. And this is the first step in programmed cell death. Now, the important thing here, and we'll be talking about this again, is that so cyclin C has a job in the nucleus, regulating transcription, but when you take it out of the nucleus and put it into the cytoplasm, it has a completely different role, activating this fission protein and causing mitochondrial fragmentation. One of the challenges we have as scientists as we're looking at this, either a stress state or a disease state, is proteins can move from one part of the cell to another and take on a completely new function. And this is what we call day job and night job. And so right now, this is the night job of cyclin C is in the cytoplasm. Now this role in apoptosis is sort of schematic here. It's not important for the details. But what happens is that the mitochondria, this mitochondrial outer membrane permeability happens, and so you get a break in the mitochondrial membrane. and out leaks a protein called cytochrome C. Now, for people who remember their electron transport chain thing, cytochrome C is an important protein just involved in making ATP. When it's in the mitochondria, that's what it does. It makes ATP, it's a good thing. However, when cytochrome C gets out into the cytoplasm, it activates a series of proteases that chew up proteins, and this is how the cell kills itself, the apoptosis effect. So cytochrome C in the mitochondria is involved in making energy. You take it out into the cytoplasm, and it's the death, Darth Vader of cell death. So why apoptosis is so important is that in neurodegeneration, neurons are exquisitely sensitive to reactive oxygen. And you remember that most neurons are post-mitotic. In other words, they've already divided and they're not going to divide anymore. So replacing them is very difficult. And if they're sensitive to cell death, it's hard to replace those things. Oh, good. So the other step we wanted to go to in apoptosis is also in aging. Too much, and just like chocolate cake, too much chocolate cake is bad for you, and too much apoptosis is bad for you, and too little is bad for you as well. And so understanding how those things go back and forth is important. And finally, we start looking at, this is now Alzheimer's, and again, you have the same theme that we see in Med13L that Alicia will be talking about later, is you see mitochondrial dysfunction, you see increased oxidative stress, you see disturbed mitochondrial genome, and this is what they see in Alzheimer's. It's the same thing they see in other neurodegenerative diseases, Huntington's, and in this case here, Parkinson's disease, where you have the mitochondrial fission, and this is the mitophagy pathway I alluded to earlier. So damaged mitochondria get taken off through this process here, and they undergo mitophagy and they get cleared, and now the good mitochondria can keep working. So what happens in Parkinson's disease is that this process doesn't work, and these damaged mitochondria accumulate, again accumulating ROS, and again accumulating mitochondrial dysfunction and neuronal death. So just to sort of finish up here, just to show you just how complicated it is, not all damage is created equal. So when you can damage cells, don't worry again about the details, cells will go through apoptosis like I just mentioned. However, a little bit less damage and the cells will undergo something called senescence. And senescence is when the cells stay alive but they quit dividing. They still produce things, but this is probably one of the topics of aging is the increased accumulation of senescing cells in tissues. So people are trying to understand now how the cells either make this decision to go through cell death or to go through the senescence process. So just to summarize, fused mitochondria are good. Life is good. You get mitochondrial function goes up. In fragmented mitochondria, you lose energy production. Mitochondrial DNA gets damaged. Apoptosis goes up, as well as the ROS. And so what we're going to be showing, what Alicia's going to be showing this afternoon, is how in Med13L cells that we've gotten, this fragmented mitochondrial phenotype is detrimental. And so just to acknowledge people, Peter Van Hesselt, who got me into Med13L, Jen, who gave us, and her crew at Columbia, who gave us those additional 12 cell lines that we got from the last meeting, which were really helpful, and Chad, who is doing better. Okay. And funding by the foundation for both Alicia and a technician, Kendall, who are now working on these other 12 cell lines and trying to determine whether any drug interventions can actually be useful to reversing this mitochondrial defect. So thank you very much. Careful.
All right. So do we have any questions for the researchers?
Yes.
Hi, I wasn't expecting a microphone. I was just wondering if it would be appropriate to talk now about funding and how much money it takes to do these types of experiments, or a little bit more detail about these institutions that are currently funding these projects, including our foundation and others around the country.
In what way?
About maybe how much it's costing or how much it will cost or how much needs to be fundraised, on a yearly basis or per program?
Sorry, thanks. In general, when we write budgets for grants, the salaries of the students are involved in that, so are the postdoctoral fellows. So those salaries are student stipends. And we figure around $2,000 to $2,500 a month per person for supplies, reagents, and for the growing cells and treating the cells and doing that. And also maintaining big equipment is usually not part of this because we usually get that through the departments. So it's very nuts and bolts for us at that point. And the foundation has helped us both with supply money as well as salary money. The current climate of the government is difficult, to say the least. So right now, it's up in the air what's going to happen. And so every university people I talk to at different universities are in the same situation. It's just not clear how this is going to finally sort itself out. So until that, we're going to try and keep going forward as much as we can. We're not going to let it stop us. So we're now actively trying to get other sources of money outside of the NIH or the NSF. So places like Mid-13L, the foundation here, and other similar types of foundations that are interested in these types of issues, we'll be knocking on their doors to keep us going.
So now more than ever?
Now more than ever, absolutely, yes.
There's a QR code on the back of the door, lanyard for these donations to continue to support, so.
Hold on, Anna. No, you have to speak into the microphone. You're being recorded.
Hi, I would like to know how different the mitochondrial dysfunction with our population is from those more classic mitochondrial diseases that have more severe symptoms and maybe even life-threatening.
That's a great question. And most of the mitochondrial dysfunctional myopathies mitochondrialopathies that they have, a lot of those involve mitochondrial DNA mutations. So a lot of times the mitochondrial DNA encodes only about 13 proteins, but they're critical for mitochondrial function. And many of these diseases have large deletions in the mitochondrial DNA. And so even though you have plenty of mitochondria there, they're dysfunctional. They can't work. They can do some of their jobs, but not the energy production jobs. And that's why many of the drugs that are out there to stimulate mitochondrial biogenesis don't really work because you can't restore the mitochondrial DNA because the deletion, once it's gone, you can't put it back, right? So what we found when we first looked at our first cell line is that in general, the mitochondria are intact. As far as we could tell, we didn't go into great depths for it, but we didn't see large deletions in the mitochondria in the Mid-13L patient samples. So that gave us some hope that if we can stop this fragmentation business that Alicia will talk about later on, and she'll talk about how we're planning on using certain drugs to help. If we can restore mitochondrial shape as well as production of more mitochondria, the DNA, I think, will allow us to now start making ATP better and doing normal mitochondrial function better. But like I said, we haven't gone deep dive into that, but it's I think, a reasonable guess that the mitochondrial DNA are probably in good shape.
Thank you for your work, first of all, Doctor. I just want to make sure I didn't mishear your beginning of your presentation. You were speaking about iron content. Is that correct? So in our current, I'm sorry, I'm lost for words. When my wife was pregnant, her iron was testing ridiculously low to the point where her birth wasn't even picked up for the first couple of tests. Was that an indication of a problem, or is that a side effect of the mitochondrial function or lack thereof?
Well, that's a good question because we haven't actually measured mitochondrial. But what I'm talking about is there's something called iron sulfur clusters.
Okay.
That iron, you need iron for normal functions of certain enzymes, but you don't want too much.
Okay.
And the mitochondria are really good at storing that in a safe way. And so mutations in proteins that are involved in that, making that cluster, the iron, when it gets released into the cell, is deleterious. It will go through this process that will generate ROS again. So iron can do this. So in the mitochondria, when the mitochondria are intact, they're better at sequestering that iron in the mitochondria. When they get fragmented and they start losing their integrity, then the iron has a chance to leak out. So, but we haven't actually measured specific iron concentrations, although that's something we were planning on looking at.
Okay, thank you.
Hi. I'm wondering if you can talk a little bit about myelin damage. You mentioned Parkinson's and Alzheimer's, and I believe that those both have like white matter myelin damage in the brain. And I know that some of the Med13L kids have myelin damage and some don't, but have the same phenotype. So I'm curious whether you've looked into that at all in terms of how the mitochondria affect it.
Well, my colleague here doesn't seem to be interested in. To be honest, I'm not sure. I don't know how myelin affects it. I would imagine the mitochondrial deficiencies would have an issue on how myelin not only forms but how it stays. Its survivability. But I can't really tell you one way or another on that.
Thank you.
I'll ask Chad later, and next conference we'll let you know.
My daughter's lactate levels have been tested a couple of times, and they're twofold. Would that probably be due to the mitochondria? And then have you heard anyone else giving any information about having elevated lactate levels?
Hello, I'm Alicia. In the corner, I will be speaking later. But so lactate serum levels, you're asking about?
Yeah.
Like in the
yes.
blood?
Yeah, in the blood, yes.
Okay.
It was blood, and it was twofold of like the highest level of what's considered normal.
Right.
It was double that.
So that could be indicative of mitochondrial dysfunction, just because of all the, to put it in like broad terms, of the different energetic processes within the cell. Lactate, and especially lactate dehydrogenase, are different ways that the cell can make energy. So it could be that these cells are trying to compensate for the lack of energy being produced from the mitochondria and are instead using lactate, or its partner enzyme lactate dehydrogenase, to kind of compensate and make energy through that pathway instead of the mitochondria. So, but we've looked at, in our first study, we looked at mitochondrial enzymes, and 8 of the 13 enzymes we looked at had reduced activity. So that was something with a skeletal muscle biopsy that we saw. So it is something that we can see like firsthand decreases in these enzymes and that kind of thing.
Thank you.